who also worked with Sarah this summer. Okay, hello everybody. I'm Brooke Keck. I was working in Dr. Sarah Hamsher's lab, and I was kind of piggybacking off of Emily Newman's master's projects and all of the work that she's been doing. So it's been a great opportunity. And this summer we were looking at determining the growing degree days of Starry Stonewort in 2 Michigan lakes. So Nitolopsis obtusa, more commonly known as Starry Stonewort, is a freshwater macroalga that is currently invasive in North America, and in Michigan is now occupying around 150 lakes. Studying invasive species like Starry is crucial because they disrupt the ecological communities greatly. And while we've been in the field, we have seen the biomass of Starry increase while other plants and macroalgae are decreasing. And we are especially concerned because Starry is spreading fast in the invasive region. And in many lakes, it's still going unnoticed, misidentified, or it's missing an official voucher. So we're still unclear about the total number of water bodies it's inhabiting. The aim of this research was focused around the phenology of Starry Stonewort. And while many studies attribute the changing of life cycle stages to biology or the internal clock, some organisms like Starry Stonewort most likely change and grow due to the environmental influences like climate and weather. And due to the variability of the changing climate and changing weather and seasonal changes, that this organism's response and their developmental rate are heavily dependent on those changes from year to year. So it's always going to be changing. So for Starry, they're heavily dependent on light availability, temperature, and water availability. And so the growing degree days aspect of this is just the accumulation of heat units over time. And in the invasive region, it also is not well known the low temperature threshold of Starry. And we hope to find the mean temperature at which the reproductive organs are appearing. So the growth and phenology of Starry Stonewort, it starts in the early summer with kind of patchy distribution and relatively low biomass. And as you move further into the summer, it starts to pillow and cloud and can grow up to over 2 meters long and reach the top of the water column. You can see it poking out at the surface. This is taken from the Marina Site. And then, depending on weather and seasonal changes, like I said, in late June to early to mid-July is when the male reproductive organs are going to start to show up. And then as you move into later August, over here is the asexual reproductive bulb that is typically present within the sediment, but can also be known to grow above the sediment. And so the male reproductive, or the asexual reproductive bulb is the means of creating new populations, as Starry is a dioecious species, so there is male and female parts, but in North America, only the male parts are present, and they are sterile. So, for this field work, this was my 2nd season working with Starry, and last summer, we had these same 4 sites. So we had, this is Pentwater Lake, and we have one site here, here, and here, and then this is Muskegon Lake with us sampling right here. So we kept those same sites, and at each site, we had, so each of the 4 sites had 2 permanent quadrats that were placed via GPS coordinates along with data loggers, water and temperature data loggers are deployed. And so at each site, we have 1 quadrat at the depth of 1.5 meters, and then another quadrat at a deeper depth, usually 2.25 meters to 3 meters. And so each week, snorkel surveys were done at every site. So I swim out to every site and every quadrat, and I take photo documentation to see the biomass of Starry growing, along with documenting if there was the presence or absence of the male reproductive organ, the antheridium. And so we wanted to go every week to make sure we did not miss when it appeared. So for the preliminary data analysis, the field season is still going on. So we haven't finished all of our collecting, and so we haven't gotten the data from our data loggers yet, but I wanted to visually show the percent cover, how the percent cover and the biomass of Starry has changed over time, along with when we saw antheridia appear. So these graphs have all of our sampling dates at the bottom, And when the antheridia first appeared is indicated by the parentheses and asterisk mark. So that is when we first saw it, and the asterisks continued as just antheridia were still there. And the stars indicate at what quadrat it was when antheridia was present. So at the 1.5 meter quadrat, we saw antheridia 7/9/21, 7/15/21, and 7/22/21. And each of these will have a picture of the shallow quadrat and then the deeper quadrat as well at each site. So this is the first site that we go to every time, and we see like a steady increase in the percent cover of starry in each of these quadrats. This was the first week we saw antheridia appear, which was the end of June, and it was earlier than we saw it appear last year. So this is our Marina Site, which I'm going to go back to this. The Marina Site is right next to the outlet of Lake Michigan, and there's lots of boats here. So there's a lot of boat traffic, and that is how starry is spread, usually on the engines, on the underside of boats, from people not cleaning them. And so we saw a big increase in starry. There was 100% cover starting around the end of June, and along with the steady increase we see, and the antheridia were present in all of the quadrats, both the 1.5 and the 3 meter, up until our last sampling date that I went on, 7/22. The next was a reference site. So this is just another residential place, and we see a little bit of varying data. And this site is interesting. We saw a kind of flip-flop of the native Cara species and the invasive starry, where Cara was closer to the shore at the beginning of the season. Starry was further out, and then it kind of changed where we saw starry closer to the shore. And so a lot of our quadrats kind of got taken over. There's a lot of native plants growing in there now, and it still is even changing. And then the last one is Muskego Site. And so as you can see, there's a lot of plants, there's a lot of starry. The reason for there being antheridia in the 1.5 meter quadrat, these two sites, and then not in the last sampling date, was we saw an explosion of starry in Muskego. I went out there one week, and it looked like all the native plants, all the other plants, aquatic plants, algae, had disappeared, and it was a forest of starry, and a lot of it was new growth. The color was light green to yellow, and I am suspecting that no antheridia were present in the quadrat because it was such new growth. It hadn't had the heat requirements or the growing degrees yet to develop the antheridia because it was so new. Okay. And this summer, I also did a lot of plant processing. So out of last summer's biomass project, we collected about 900 plants. And so I processed about 600 of those, where I'm making labels. We put them in folders, or we make a voucher, and they're going to be sent to the herbarium at the New York Botanical Garden. This has been a lot of very detail-oriented, time-consuming work, but it's been great because I've learned a lot of plant identification. I've learned, time management. I've worked on detail orientation, and it's been a great experience to work on all summer. Okay. And so I just want to wrap up by saying thank you to AWRI. I've been here for close to a year and a half now, working, and it's a great environment. It's given me a lot of opportunities. So I want to thank everyone at AWRI. And a special thank you to Dr. Charlotte Partridge's lab and Mike Sanders. I got to go out in the field working with the HWA project. So it was nice I got, aquatic field work and terrestrial. And then lastly, of course, Dr. Sarah Hamsher, who could sadly not make it here today, but she is my academic advisor, she's a mentor, she is my supervisor here, and she has given me nothing but support and great ideas and experiences throughout this whole process. And to thank Emily Newman for letting me piggyback on her project, help out last summer, even though I had no idea what was going on, to now where I feel like all I want to do is talk about starry, and I want to tell everyone about it. And thanks to Kelsey for helping me out in the field also. So that was a really big help, and it's fun with all of our long car rides. Okay, so that is the end of my presentation. Thank you. Questions for Brooke? Yes.
Do you think that the water levels from last year, this year, caused the antheridia to form
think it has something to do with it. I think we saw at the beginning of this year, the water was really low for a long time. Like we showed up and the water level, we were like, whoa, this is way down. And then we got at our first site at the Riches, the management site, he put out a meter stick so we could track water. And I think just about when water level began to rise, shortly after the antheridia grew, so maybe more light was penetrating into the water column, giving the necessary heat and light requirements needed to grow the antheridia. So yeah, possibly, yeah.
One quick question, in terms of the asexual bulb, is that the only way it can reproduce asexually? Like, does that mean to kind of break off and form a new individual, or does it fragment where each fragment or
I actually don't know. Emily could probably answer that really quickly, how the
the nodes to balance branches off.
Yeah.
No, and so the best means for management right now, people do a lot of mechanical manual harvesting, there's chemical harvesting, but a lot of what should be doing is just prevention. And so we want to prevent it from even starting to like fragment and grow via the bulbs. So.
Any further questions?